laptop, but it's like the wrong way. So it's got like the male VGA and the female HDMI, so I have to get another one, unfortunately. But I will get one, and then I won't have to click two laptops. So yeah. Yeah. So we're kind of reviewing some of the stuff we went over in a rush at the end of last class, just to make sure that we're all on the same page. So when we talk about biomolecules, what that concept means is these are the classes of molecules that we're going to work with frequently in biology. So you've probably seen at least some of these categories before. So we're going to be talking about carbohydrates, lipids, proteins, and nucleic acids. So an example of carbohydrates is glucose, which we find in just both plants and animals in different forms. And those different forms of glucose are going to be bonded in different ways, and it's going to impact their function a lot. So we're going to talk about that first. They're going to move on to lipids. We're mainly going to focus on the lipids that make up the membranes of cells and how those function and how those are going to change in different conditions. Proteins, we're going to talk a lot about the way proteins fold and unfold, especially in different environmental conditions and how they can impact protein folding. And then we're going to talk about nucleic acids. So nucleic acids, an example of that is DNA, which I'm sure you've heard of. So DNA contains a lot of information that's vital to cell function, and we're going to talk about how DNA accomplishes that. So carbohydrates, so they're made up of carbons, oxygen, and hydrogens, just like this. So their main functions in plants and animals are to serve either as a fuel or as a structural component. So glucose actually also makes up the outside of plant walls as cellulose. So just one carbohydrate unit is called a monosaccharide, but we're going to get a bunch of these connected to each other. And if there's just two connected, that's what we're going to call a disaccharide, which is a really common molecule to store energy that we can move around cells in animals. Or we can have polysaccharides, where it's more than two of these monosaccharides linked together. And so an example of that is like cellulose. In cellulose, you have a long chain, like thousands of these monosaccharides linked together to form one rigid structure. So when we have, for polysaccharides, when we have more than two joined together, they can be joined together in different ways. So up top, we have a starch, which is more organized than a glycogen down here. So the glycogen looks kind of randomly linked together, whereas the starch is not. It's a little more orderly like this. And that's going to be a function of these different bonds between the individual monosaccharides. And so plants and animals are going to have different forms of these polysaccharides, and these different forms are going to dictate their function. So even though the molecular structure is the same, there's all the same atoms in here, the way they're arranged relative to each other is different. And that little minor change actually makes a really big difference with the way these polysaccharides, these molecules, function in biology. So it's really important to pay attention to these small details with bonding to make sure that you understand how different molecules are going to be working. So you don't have to memorize this. I just want to show you an example of the different behaviors that we can see. So polysaccharides are important as an energy storage mechanism because they're so big that they're easy to contain within a cell. So if you have just individual monosaccharide here, just an individual glucose molecule, it moves around a lot quicker inside the cell, which can be good sometimes, but if you're looking for like a long-term energy storage molecule, then that's not ideal. So you want a bigger molecule like a polysaccharide. So this is different. This is even more organized than we saw in the starches. So you have these long parallel chains of polysaccharides, and in between you have these hydrogen bonds. That's what these dotted lines signify. So these hydrogen bonds are going to make these parallel chains kind of stick together like this. And so you can get one long fiber. And these long fibers give plants their structure. So we can't eat cellulose and digest it, but certain animals have microorganisms like bacteria in their gut that can break these bonds apart into a form that's then usable for animals. So cows, I think, actually have a bunch of these microorganisms in their stomach, and so they can eat cellulose like grass, and the microorganisms break down the grass into a form that's digestible by the cow. Like, we don't have that, so if you just ate grass, then you wouldn't get any nutritional value. So why does it So this is not something that gets broken down. It's just like dietary fiber is basically just like a physical mechanism to like help the gut function properly. So you don't actually, it's a good question though, you don't actually break these bonds apart in fibers when you like eat them, but just the fiber in your stomach and in your digestive tract helps it function better. Yeah, good question. So yeah, if we compare these, glycogen, this is something that we can digest, and this is cellulose, something that we can't digest. So all of the atom, there's all the same atoms in both cellulose and glycogen, but the way that the bonds are arranged between the glucose monosaccharides is different. So here you can see that the oxygen is always on the bottom, whereas here it alternates back and forth. So when it alternates like this, it's easier to pack it more tightly to get these long parallel chains, whereas when it alternates, you get kind of more of a zigzag. And even that small change affects whether or not we can digest something. So again, these are carbohydrates. Individual one called a monosaccharide. A long chain of them is a polysaccharide. Just two is a disaccharide. So we're going to see these more moving forward, but just want to expose you to some of the molecules, biomolecules that we'll be talking about as we move forward. So lipids. Lipids are different than monosaccharides. So these are hydrophobic or water insoluble, which we could predict from looking at the molecular structure here. We see a lot of C and H bonds, and we know that carbon and hydrogen have similar electronegativities, so these will be nonpolar. So we'll talk about moving forward the difference between saturated and unsaturated. But again, it's just a small change in whether or not there's a double bond or single bonds is going to change whether or not something is a liquid or a solid at room temperature. Lipids are used mainly for insulation as like a long-term energy storage molecule, and also as a structural molecule on a cellular level. All right, so we have a special type of lipid that we're going to be talking about moving forward when we talk about membranes, called triglycerides. So what we have here is 3 of these chains. These are fatty acids, and one head group, which we call glycerol. So the fatty acids are just a bunch of carbons with hydrogens attached to them, and the glycerol has these carbon and oxygen bonds, as well as carbon and hydrogen bonds. So which do you think is going to be more polar, the glycerol or the fatty acids? Yeah, so the carbon and hydrogen have a similar electronegativity, so this bond will be nonpolar. So these are all going to equally share the electrons, all these carbon and hydrogens, whereas the carbon and oxygen is going to be a polar bond. So that's going to make the glycerol more polar. So we have a polar glycerol head and nonpolar fatty acid tails. So what do you think would happen if this was in water? Which side would want to be near the water? Which side would want to be not near the water? Great, exactly. So fatty acids, hydrophobic, nonpolar, they want to be away from the water. Glycerol, polar, wants to be near the water. So that's going to give us an interesting structure. Oh, sorry. Do you want to go back? Yeah, exactly. All right, so when we talk about triglycerides and fatty acids, we should talk about saturated or unsaturated fatty acids. So actually I kind of squashed a good graphic here. So let me change this real quick so you can see it. Sorry, I went through and took out the animations because I thought they were distracting, but I forgot that some of them had things under them. Sorry about that. Okay, so this is a saturated fatty acid on the top, and this is unsaturated. So with the unsaturated, we have these double bonds. The saturated, we have no double bonds. So you can think of saturated to mean like saturated by hydrogens, like that's the maximum possible number of hydrogens. Because when you have these double bonds, that means the carbon can only bond to one hydrogen and not two. Because it can only make 4 bonds because it wants to fulfill its octet rule and it only needs 4 electrons. So it's only going to bond to 4 things. Here, each carbon is bonded to 2 carbons and also 2 hydrogens. So it has more hydrogens up here and fewer down here. So unsaturated means it doesn't have the maximum possible number of hydrogens. It's not saturated by hydrogens. Does everyone see the distinction in saturated versus unsaturated? So with saturated, when you have the maximum number of hydrogens, you get these more parallel chains of fatty acids. So as a result of this structure we see up here with more hydrogens, you get a more parallel structure. Whereas with unsaturated, you get this bend in the chain where you see double bonds. So you can see that here. And that's the reason for that we're not going to cover in this class, but in your chemistry classes, you'll go over what makes that difference.
Not to go like too far back, but what just like really, really trips me out is that all these, their octets are all complete.
Yeah.
All the way around, and even any of the other molecules. So like, is it just due to small electronegativities, like the reasons why they would ever want to combine if they're always complete?
Well, so these atoms only have a complete octet because they're bonded. So that's why they. Sorry, I don't understand your question.
I guess like if you look at like carbon at the end,
yeah,
it's connected double bonded to oxygen and the HO.
Yeah.
Like, is that? But that's still going to accept a connection from another chain.
Right. So if you wanted to attach like one of these fatty acids to like a glycerol for making a triglyceride, it would actually change this bond structure to kind of attach it to something else. So that's actually kind of going to be a good segue into something we'll talk about towards the end of this chapter and in future chapters. When cells change the structure of these biomolecules to do kind of useful work, whether it's breaking something down or clearing out waste, they're going to be changing the bond structure here. And as they change the bond structure, they kind of change the function of these different biomolecules. So the bond structure that we see here isn't always going to hold true as the cell works with these fatty acids. So they can change them and modify them to their needs. But to do that, it kind of costs a lot of energy. And so they want to do that as efficiently as possible. But so all of these atoms have fulfilled their octet rule, and that's why they're all bonded together. Otherwise, they wouldn't want to stick near each other. They would want to kind of float around on their own way and do their own thing. But since they're bonded together, they're happier fulfilling their octet, and they're happier being together. But when we have these unsaturated fatty acids, we get these, they call them like a kink in the chain. And that's going to change whether or not it's solid or liquid at room temperature. So oils have like kinked fatty acid chains. So these are unsaturated. They don't have their maximum number of hydrogens. Whereas fats are saturated, at least in a biological context when we talk about fats and oils. Kind of different when you talk about it in everyday life. But if we use these kind of classifications, we have fats that are solid at room temperature and oils that are going to be liquid at room temperature. And that's a result of these chains being kinked or not kinked. So when you have chains that are parallel like this and straight, they're more likely to want to be close to each other and not flow around as much. They're going to pack more tightly and they're going to have more of a structure. When we have chains that take up more space like this, it's like some guy who sits on the subway with his like knees all the way apart, so you can't get as many people there. So things are not going to sit as close to each other, and they're going to flow around a little bit more. You're not going to have as much of a attraction between the two of them.
So like your example of butter, since butter can melt, like some liquid melted, would the chain still be straight or would they bend?
That's a good question. So if you have butter, so butter is like a solid room temperature, and you heat it up, the chain structure is still the same, but you've added energy into it by increasing the heat. And so even though, sorry, I was pointing the wrong thing. Even though usually they'd want to sit near each other, as you increase the energy, you basically make everything shake around a lot more. And so it doesn't want to stay as close to other, butter or other fat molecules. So yeah, as you heat it up and you increase the temperature, just like kind of ice that would go from solid to liquid, butter does a similar thing for similar reasons.
So unsaturated fats are like healthier for you from like a nutritional perspective, and is that because the fatty acids should be different?
That's a good question. I don't know why that is. I would guess that it's because saturated fats, since they're a solid, maybe are more likely to kind of get stuck in your blood vessels and cause like plaques to form, and unsaturated ones are more likely to flow out and not get stuck. But is it like a general statement that unsaturated fats are more healthier than saturated? So, okay.
I'm guessing that the oils are also easy to work with, like as far as changing them to do a certain function in the body because of the kinks that they have.
Right. Yeah. My nutritional background is pretty terrible, unfortunately.
It's just like they use fish for like peels for the skin.
Right. Yeah. Yeah. So that's a good kind of segue too. So unsaturated fats are going to come primarily from plants and fish, like fish oil or like peanut oil or olive oil, whereas saturated fats are going to come mainly from mammals. And that's going to be like lard or like insulation that forms under the skin to keep mammals warm.
Sorry,
No, it's
but like coconut oil, would that work the same with saturated fat?
that would be like an unsaturated fat, coconut oil. Oh, sorry, is that a solid room temperature?
Well, in room temperature, salt is
Right.
okay, but if even gets like this like a little bit warmer, it starts to melt.
Yeah.
Right.
Yeah.
So maybe so this should be kind of a general rule, like primarily from plants and fish. So maybe coconut oil is a little more saturated than most plant oils, and maybe that's why it's like solid, just right at room temperature. There's always it's hard with bio. There's always like little exceptions to every rule. So if you take a lot more bio classes and you look back at these slides, you're gonna think none of this stuff is true. But these are like general rules that generally hold true, but there's gonna be exceptions to everything. Any other questions? All right, so when we talk about long-term energy storage, we see it a lot in small birds, like migratory birds, because they don't have food for long stretches of time. So they need the most efficient energy storage by mass, because they want to stay really light so they can fly, and they need energy for a long time because they have like very long flight paths where they can't stop for food. So fats actually contain a lot more energy than glycogen does, which is a type of carbohydrate that we talked about earlier, that we see in like if you eat meat and you eat like a mussel, that's going to be full of glycogen. So it has more energy by mass. Like for 1 gram of fat has a lot more energy than 1 gram of glycogen, but it takes longer to release that energy. So you wouldn't want it if you're just kind of walking around throughout the day, or you have to sprint all of a sudden. That's not a good use for fat, but it is a good use for glycogen. So birds have a lot of fat in them, especially migratory birds. And now, seal. Is seal or a walrus?
A
walrus. Thank you. Should have known that. So walruses have a lot of fat as insulation. So they have to keep their bodies warm in really cold temperatures. And fat has a lot more insulation than other types of biomolecules. So even though it takes a lot of energy for a walrus to build up these thick fat layers on the outside of its skin, and it has to carry them around throughout its entire life, it needs those to keep the rest of its body warm enough to function properly. So you can kind of sort of see how you can look at biology, especially animals like migratory birds or walruses, and the way their structure is specially designed for their function. Kind of like a chicken or the egg. Like, it's hard to say what came first, but they all have very specialized roles that they have to do as efficiently as possible in order to survive. Okay, so we talked about triglycerides. So what we see up here, we see a fatty acid tail with a glycerol head group up here. And now one type of triglyceride, or one similar molecule that we'll talk about a lot, are these phospholipids. So they just have two fatty acid tails and then this phosphate group. So this phosphate group, so since phosphorus and oxygen have very different electronegativities, do you think this is going to be, more or less polar than the fatty acid chain that replaced? Sorry, more polar. Does everyone see that? So since phosphate and oxygen have a big difference in electronegativity, that's going to be a polar, this is going to be a polar group, this phosphate group. So this area is going to be more polar than the fatty acid chains. So this is going to make it more likely or less likely to dissolve in water? More likely, yeah. So the glycerol head group and the phosphate group are going to be hydrophilic, and our fatty acid chains are going to be hydrophobic. All right, now my favorite part.
So you said
Well, so that's a good question. The phospholipid has a hydrophobic region and a hydrophilic region. So this whole thing is a phospholipid, and it has these hydrophobic fatty acid chains and these hydrophilic, the glycerol group and the phosphate group.
Okay.
So a molecule, like a big molecule like these, can be both hydrophobic and hydrophilic. It can have two different regions. And when it does that, it'll have a setup like this. So these are the hydrophilic head groups. These are the hydrophobic tails. And often we'll get bends in the tails like this, because do you think this is saturated or unsaturated? Unsaturated, because there's a double bond here. So that means these carbons only bond to one hydrogen rather than two. So it's not saturated with hydrogens. So when we have these phospholipids, when we put them in water, they have this really interesting behavior where they'll put their tails together, their hydrophobic tails together, to keep them away from water, and they'll stick their polar head groups out, away from the water. The recording's going to be off now, sorry about that. So we have the hydrophilic head group and the hydrophobic tails, and the hydrophobic tails are pushing against each other to keep the water away, to make sure there's very few interactions between the hydrophobic area and the water. And the head groups, which are hydrophilic, are going to be pointed out towards the water. And that's going to make this bilayer here, is what we call it. So you have a layer of phospholipids on the left and a layer on the right, and they're both going to be pushed up against each other to make a bilayer. And this bilayer is what surrounds cells and surrounds organelles within cells.
What's inside the bilayer?
Really just the tails. No liquid is in there.
No empty space?
Well, yeah, so that's a good question. So they try to pack them as tightly as possible to have very little empty space.
Would the empty space just be a vacuum?
No, well, so as these tails are pushed up against each other, they're going to be sharing electrons between them a little bit, and the charge between them. So there won't really be a vacuum. They're either going to be packed very tightly. I guess there maybe be very small amount of water, but mainly it'll be very like a non-polar area. I wouldn't think there'd be a vacuum. I'm not sure what would really be like in the space between the chains. That's a good question though. As a general rule though, you could think that there's like no water in between.
Yeah.
But maybe there's just some that gets trapped in there, but it's really mainly just the non-polar tails. So we see plasma, or these membranes, we see them, they encapsulate entire cells, and they surround individual organelles. And so what they do is they allow the cell and the organelle to have a very specific environment because it's kind of encapsulated and totally closed off from the rest of the cell or the area outside of the cell. And so that means what happens inside the cell, or it happens inside one of these little organelles within the cell, can be controlled. It's like if you had a house, you would probably want walls in your house to keep all your stuff inside and keep people out. So that's kind of like the walls of the cell, just kind of keep unwanted things out and keep the wanted things inside.
It's just water inside too?
Yeah, great. Well, but there's different things dissolved in the water inside, but yeah, it's water based inside and outside. And most of the organelles are like water based inside too. So we'll talk about the water based substance that goes inside of the cells later. It's actually like a really viscous thing, kind of like syrup is really viscous, like it flows really slowly. So it's just so packed with things in there inside of the cell that it's like really thick like that. Yeah, exactly. But yeah. Sorry, I didn't want to. No. Thank you for saying it. Sorry, I have a tendency to just like talk about everything that's related to what we're talking about, but I'll try to stick in the chapter that we're at. But if you have any questions, feel free to ask me. Everyone excited about plasma membranes? They're really cool. I just think it's interesting that these. Oh my gosh. This small difference, how we have a hydrophilic head group and a hydrophobic tail group, if you just put enough of them in water, it spontaneously forms this structure, just to minimize the interactions between the hydrophobic areas and water. So this is just something that naturally occurs because these molecules were synthesized this way.
Kind of looking at the picture over there, are the two hydrophobic tails bonded to each other, or I know there's a gap up in the water.
No, so I did say bond. The term bonded we usually reserve for ionic and covalent bonds, and that's what we should really stick to using it for. But there's interactions between these tail groups that keep them close together. So when you have these long hydrophobic chains, they interact with each other and they do like to stick near each other, like we saw with saturated fats that form a solid. So the more parallel those chains are, the more they like to stick together. And so you can control that behavior a lot with the number of double bonds that you put in there. So as cells are making biomolecules that they need to function, they're going to change the structure with the number of double bonds or single bonds that they'll put in to these phospholipids.
But looking at the picture directly above that, I'm wondering what's keeping the tail of one attracted to the tail of the other one.
Right, up here?
Yes.
Yeah. It's because, okay, so you have these long chains and they have electron clouds that are kind of equally dispersed throughout the entire chain. But over time, they're going to kind of like jiggle back and forth just because if it's warm enough, everything kind of shakes back and forth. And when you do that, you're going to have positive and negative regions that are going to form momentarily. That's going to cause like a weak attraction between the two chains. That's going to keep them. We don't have to know that for the quiz, but just like as background, that's why chains are kind of attracted to each other. Does that answer your question? So you can ask me after class, sorry. So there's a lot of membranes between organelles and outside of cells that we're going to be talking about. So we're going to do a quick activity, and then we're going to take a break after that. So in groups of 4, just get together and talk about what you think would happen if you put a cell with a plasma membrane, like the membrane bilayer, in something that's less polar, like ethanol rather than water. So ethanol is more hydrophobic than water is, so it's going to change the way that phospholipids arrange each other. So just get in small groups and talk about what you think might happen. If you don't know, that's fine, but just try to brainstorm something you think might happen. And we'll get together in a few minutes. Okay, great. Sorry, earlier today the keyboard on my laptop died, and now it just randomly like will stop working. So it just has momentarily stopped working. So hopefully that doesn't turn into a bigger problem. So the third class biomolecules that we're going to talk about. So we talked about carbohydrates and lipids. Now we're going to talk about proteins. Proteins are, in my opinion, a little more exciting because they have a wider range of activities that they can do. So proteins always have this common structure, but this R group, this area here, which just stands for like rest of molecule, is different in different amino acids. So just like in carbohydrates, we talked about monosaccharides as like the smallest building block, the smallest unit. In proteins, we're going to be talking about amino acids. And so each amino acid has a different R group. And that's going to give it different behavior. But the rest of the molecule, which you don't have to remember the names of this, you just have to know that the R group is different amino acids. The rest of the molecule is always going to stay the same. So the rest of the molecule is just like, kind of like a platform just to have the R group on there. And the different R groups are going to give different amino acids different properties. And as you arrange the different amino acids, you're going to have proteins with different properties. So in total, there's 20 amino acids. Again, don't memorize all of these. But each amino acid has a different R group like we can see here. So the area that's shaded in light blue here is a different R group. And so we're going to talk about what some of the different R groups could look like and what that would mean for the amino acid. So if we look at these amino acids, so all of these R groups are mainly carbon-hydrogen bonds. So do you think these amino acids are going to be polar or non-polar?
You said carbon-hydrogen?
Yeah.
It's polar.
So carbon-hydrogen have a similar electronegativity. Non-polar, yeah, sorry, now I got mixed up myself. So carbon-hydrogen have a very similar electronegativity. So all of these amino acids, which have mainly carbon and hydrogen bonds, are going to be non-polar. So you can see that each of these amino acids, the top area, these top groups are always the same, but this area, the R group, is different in each one, and that's why they're all named differently. And so this group, these are all polar. So these are not going to want to be in water. But if we look at these ones, so we have carbon-oxygen bonds, nitrogen-hydrogen, though it's a very big difference in the electronegativity. Yeah, carbon-oxygen. So these bonds, are they going to be polar or non-polar? Polar. So these amino acids are going to, what, want to be in water or not in water? Want to be in water. Great. I keep repeating this point because it's really important that we all understand the connection between electronegativity, polarity, and whether something's hydrophilic or hydrophobic. That's going to be something that we build on a lot throughout the course.
So the only thing that makes a molecule hydrophobic or hydrophilic is polarity, like if it's polar or non-polar?
Yeah. Yeah.
So if it's polar, it likes water?
Yeah, exactly. Yeah. Because water is also polar. Yeah. So yeah, good. Thank you for clarifying. And so as we have different amino acids with different R groups that are either polar or non-polar, and we're going to arrange these amino acids to give proteins a unique structure. So we can have a protein with a long stretch of polar amino acids and then non-polar amino acids, or vice versa, or like kind of mixed randomly. And we're going to start to look at how that arrangement is going to affect the function of these different proteins. So when we talk about proteins, we talk about different levels of structure. The first level of structure is called the primary structure. So the primary structure is just the sequence of amino acids. So proteins can be hundreds or thousands of amino acids long, and the sequence of the amino acids in that protein, like the first one, the second one, the third one, that sequence is the primary structure. So these amino acids are bonded together in what we call a peptide bond, and they form a sequence of amino acids. And so we're going to talk about that as the primary structure. So a good analogy that we found useful in previous classes is the primary structure is like the order of letters in a paragraph. Like that alone doesn't really give us a lot of information. We like to look at like a larger structure like words or bigger concepts when we read, not just, you know, the exact order of the letters. And that's similar here. If we just only think about the primary structure of these proteins, you don't really have enough information to build on. You want to look at kind of longer range patterns or interactions, which is what we'll talk about moving forward. So primary structure is the sequence of amino acids. Secondary structure is how that protein kind of folds in on itself. So this is like the what we call the local structure. So it's the structure on like a smaller length scale, like just a few amino acids that are interacting with each other. And so we kind of have these alpha helices, we call them, or beta sheets, but there's just these long range structures that are either these coils or sheets that are determined by hydrogen bonds between different amino acids. So if you have two polar amino acids that are maybe like 10 amino acids away from each other, they can fold up, make the whole protein fold up so they can link near each other to form a hydrogen bond, which is what we have here with the alpha helix. So you actually see a hydrogen bond between these two amino acids here, and they can be pretty far away from each other. And so that's going to be a product of the exact amino acids that we see in that particular protein. But again, so we have the primary structure, which is just the sequence. Now our secondary structure is the way it folds or coils in on itself over a small length scale. And then we have, more information on secondary structure. So we have hydrogen bonds, usually that make up the secondary structure. And so what were hydrogen bonds again? Does anyone want to review that for us? Maybe, sorry, someone else want to raise your hand? Sorry, just you've been participating a lot, which is great, but I just want to share I appreciate it though. Yeah. Sorry, what's that? So I think you're describing what a hydrogen is. So a hydrogen is a one proton and one electron. But a hydrogen bond is when you have, like if you have an oxygen bonded to a hydrogen, oxygen is a lot more electronegative than hydrogen is. So it gets like a lot of the electrons. So this is partially negative, and then hydrogen is going to be partially positive because now it just has its proton in there and no electron. Yeah, exactly. So if you have like areas like this, and then you have another amino acid that has a similar structure. So we have the partial negative and partial positive. The hydrogen bond is this interaction. So it's a weaker interaction between a partial positive hydrogen and a partial negative oxygen or another molecule that is bonding with the hydrogen. But in between two different amino acids, we can have these hydrogen bonds that form to create our secondary structure. So primary structure is the sequence of amino acids. Secondary structure is kind of the smaller structure that's a function of the hydrogen bonds. And then our tertiary structure is the whole three-dimensional folding of a protein. So like I said, a protein can be hundreds or thousands of amino acids long. You have the primary structure of the sequence, the secondary structure, just these small hydrogen bonds like between amino acids that are near each other. And then you have this tertiary structure that is the entire protein in its three-dimensional shape. So the tertiary structure is like a three-dimensional shape of a protein. And the three-dimensional shape of the protein is what's going to dictate its function. So you can have covalent bonds, which is what happens here. So if you have two amino acids with sulfurs on the end, they can actually bond together to form a covalent bond. And so if you have this covalent bond, that means this loop is going to have to stay, kind of looped around like that because you want these areas to be near each other. So if you have a long string and you take two ends of it that are far away and you like glue them or tape them together, you're going to get a loop that forms permanently. That's like what happens here. You can have more hydrogen bonds. You can have what we're going to be talking about a lot, this hydrophobic effect. So the hydrophobic effect is like what we see in plasma membranes, where we see the nonpolar tails that want to stay near each other and exclude water. That's what you'll get here. All these nonpolar amino acids here are going to want to keep water out and just stay close to each other. And that's going to make a structure form like that too. You can have ionic bonds between amino acids that are likely to form ions. But you can have a lot of structure that occurs in these proteins, and this is all a function of the primary sequence, the primary structure. So if you can control, the order that you bond these amino acids together, you can make different tertiary structures. And as you make different tertiary structures, you make proteins that have different functions, and they can interact with other biomolecules and make really interesting things happen.
So just to make sure I understand right. So essentially, you have a sequence of amino acids which form your primary structure. They kind of fold around to form your secondary structures, and those fold around things loop around to form your third structure.
Yeah, that's a good way to think about it. Yeah. So the most important things to keep in mind: tertiary structure is like the entire 3D structure of the protein. So if you only remember two things, remember primary structure is just the sequence of amino acids. Tertiary structure is the entire 3D configuration of the protein. And the tertiary structure depends on the primary structure.
So could you kind of look at it like the tertiary structures, like the whole building, maybe, and the secondary structures, like the rooms of the building, and then the primary structures, what the building is made of, wood.
Yeah, that's a great way to think about it. Yeah, so if you had a building with no nails, so your primary structure had no nails in it, then your building would not have a great three-dimensional structure. It would just kind of be collapsed on itself. Whereas if you had one with a lot of, a material able to structure, then you could have a strong, tall building. Another question? I thought someone said that. Yeah.
Can it be like a liquid form, moving, becoming solid?
What's that? Sorry.
Can it be like a liquid form, changing and becoming solid?
I don't think we'll ever see liquid proteins, just because. That's a good question. I think there's just always going to be enough interactions between proteins and amino acids to keep them near each other to prevent them from flowing like a liquid would, and so always be solid. So that's a good question. Not like fats or lipids where we can see those as liquids. Proteins we won't see in liquid form. Maybe if you heat it up enough, maybe you'd see one. Good question. Sorry, I'm not really sure, to be honest with you. So when we talk about tertiary structure, so like I said before, to do a very specific function, a protein needs a very specific structure. It's tertiary structure. And it has that tertiary structure because of the primary sequence, primary structure that it was made with. But when we heat up proteins or change their pH or other things that we're actually going to talk about in the lab, either today or on Thursday, as you change the temperature of the pH, you can denature the protein. And what denaturing means is basically means you've broken the protein. It's no longer in its structure, its tertiary structure that gives it the function that it needs. So an example is if you have eggs and you're cooking eggs, you're just increasing the temperature. You take these coils of proteins that you had before when the egg was laid. As you heat it up, you denature these proteins, and you actually form a bunch of cross-links now between different amino acids that have these sulfur molecules on the end. But what's important is that you took a water-soluble protein in the egg like this. So these are proteins that were dissolved in water, and as you heated it up, you denatured these proteins, and you formed an insoluble, something that's just not dissolved in water, like not the yolk, but what's the white part? I was just called the white. That's why I couldn't. Thank you. I can't think properly when I'm talking. It's not very convenient for me. So as you heat up eggs, you denature these proteins. So this is an example of denaturation of proteins. You get kind of a similar effect when you change the pH as well, which is why buffers are so important. So you keep your proteins from denaturing.
Will it also reduce the protein content?
So you still have all of the same amino acids. They didn't go anywhere. You could have changed them to a form that you can't digest anymore, I think, perhaps. I think eggs, I would guess, isn't an example of this. I think the nutritional content stays the same, but I can imagine an example where the denaturing of the proteins kind of decreased. I don't know if anyone else knows a lot about nutrition.
What if it's hard in the shell?
What's still the same phenomenon? I mean, so all of the amino acids are still staying in the egg, regardless of how you cook it, I think. I mean, unless I could see that not being true. But you could change, by changing its structure, you could change whether or not you can digest it. So, I mean, I could see that being the case. Like, I bet if you burn meat really bad and you eat it, it probably doesn't give you the same nutritional content. That makes sense then.
Cook the hell out of it, you're really not going to want to do it.
Yeah, that's true.
Yeah, no, it's not going to have the same value.
Yeah, there's like two ways to accomplish that. But so what's important from this slide really though is this concept of denaturing of proteins. So denaturing just means like breaking them. They just don't have the same function that they did before because you changed the structure, either by heating it or changing the pH. And that's why in biology, we have buffers to keep the pH constant. We have insulation in our body or other ways of regulating the temperature to keep the temperature exactly the same. And so this phenomenon of denaturing is part of the reason that biology is so complex, because cells always have to keep the conditions very constant and consistent, and they cost a lot of energy to do that, to keep the pH the same and the temperature the same, because there's always going to be like little variations. And so cells and animals use a lot of, and plants as well, use a lot of energy to maintain this level, this constant optimal level of temperature and pH, and salt content and other things like that. Because if they don't do that, then all of their proteins are going to get denatured, and proteins are really important for making things happen, for getting things done, for processing other biomolecules or breaking things down or building things. Those all depend on proteins. And so if you let the temperature or the pH change too much, then your proteins can't do their job, and then the cell or the animal or the plant is going to die. So that's why they have to invest a lot of energy into forming these cell membranes to keep their environment constant, or into making fat layers or pumping blood back and forth. So there's also quaternary structure. So primary structure was the sequence of amino acids. Secondary structure is this local structure formed by hydrogen bonding. Tertiary structure is the entire three-dimensional shape of a single protein. And quaternary structure is how multiple proteins are going to come together to form a bigger complex or like aggregation. So this is hemoglobin. This is a molecule in your blood that we're going to talk a bit about in Tuesday's lab and Thursday's lab. And this is what's going to carry oxygen around. So this is 4 different chains, so 4 different proteins that are stuck together to form a quaternary structure. So quaternary is how multiple proteins are going to interact with each other to form a bigger structure that can do something useful. All right, an overview, and we'll probably just stop after this and finish the rest of the chapter on Thursday, so we're not rushed. So we have our primary structure, which is just the sequence of amino acids, and you can think about that like as letters. So if you have a paragraph or a few paragraphs of text, just the individual sequence of letters doesn't really tell you a lot about what's written. If you just said write off the letters to someone, that wouldn't really give them a lot of information. It's the same way with the primary structure. The secondary structure are like individual words, so these are just like smaller structural components. The tertiary structure, this is the entire three-dimensional structure of a single protein. That's like sentences. So this is where you start to really get some meaning out of the text that's been written, because you have like a whole sentence. It's like a whole idea. Same with the tertiary structure. that's the entire three-dimensional structure of an individual protein, so it will have a very specific function. And then you can have a quaternary structure, which is multiple proteins that are linked together. So quaternary structure, like this would be like hemoglobin, where you have 4 proteins that are all stuck together, and they have an even bigger structure and function. And that's like the paragraphs in text. That's like a lot more information. All right, so we'll just do this real quick. Just a quick example. So these are 2 different blood cells. This is a healthy red blood cell, and this is a red blood cell of someone with sickle cell anemia. So the difference is just one amino acid that's difference between a normal hemoglobin and someone with sickle cell anemia. So it's just this one small change in the primary structure of these proteins, but it makes this huge change in the overall shape of the red blood cell. So these small details can be really important. So when someone has sickle cell anemia, their red blood cells, they don't carry oxygen as well, and they don't flow as well through blood vessels. They kind of can get clogged up and jammed in these smaller vessels, whereas healthy blood cells can flow more freely. So this happened because rather than having this glutamine up here, we instead have a valine. And so glutamine is polar, so it has this CO group up here on the upper left, whereas valine doesn't. And so without this polar group on this one amino acid, you get an entirely different structure. So with a different primary sequence, even on a really big protein like hemoglobin, which has actually 4 proteins that are all stuck together, if you have just this one small mistake, you can get a totally different structure, a totally different function that makes someone a lot less healthy, a lot less able to, run and exercise heavily.
Now, if you're a carrier of it, if you don't have it, you can't carry it in your system. Would it just be would it have like part of that polar?
So that's a good question. So you, if you're a carrier but you don't have it, you still have the normal sequence, but your DNA makes you more likely to give birth to someone that would have this different primary sequence. So your proteins are still the same, but your DNA would actually be different, which is what we'll talk about on Thursday. That was a good question. All right, that's it for today. Thank you for paying attention.